Hey everyone. Today we are going to be talking about cephalopods. In my opinion, one of the coolest group of inverts to work with. Uh, Fascinating species, a group so interesting behavior wise, training wise, you name it. A ton to process here. And I think arguably they're probably, at least some of you, maybe your most favorite invertebrate species out there. They are for a lot of people. They're super entertaining, amazing to watch at your aquarium, and yet we're still within the mollusk category here. We're talking really smart snails, basically, and that weirds a lot of people out. But they definitely deserve their own day in the sun, if you will, as far as everything they've accomplished. And while hopefully a lot of you are interested in them already, I think today's lecture is really going to dive into what really sets these animals apart from everything else. So let's get started. Again, and I know I preach this every class, you're probably sick of hearing it, but I'll do it again. When you do taxonomic hierarchy for your other courses, you're always starting like maybe class order usually is where things start changing things up. We're changing all the way up here in the phylum Mollusca. That again, and again, I know you're sick of me hearing it from me, shows how much adaptive radiation and how much these species have truly branched off and been able to do their own thing throughout the course of time. So within our group of cephalopods here, we're at the class level now. We're talking a couple groups. Again, this is one of those weird times where, thanks to cladistics and the taxonomical hierarchy we use nowadays, really only one branch gets moved around here, and that's the nautilids. As far as how we're going to break down today's lecture, we're going to start with the nautilids and move to the rest of the gang shortly thereafter. Probably everybody's familiar with squid, cuttlefish, definitely. Maybe nautilids as a gift shop trinket, but nautilids don't ring a bell to the non-zoo people of the world. And then the last one is, of course, octopus or octopi. This one has confused people for ages. I'm here to bring you the truth, the hard-hitting news. If I see multiple octopus, what do I call them? Well, that's up for debate. Actually, it's not, but it's up for your interpretation. The very common thing is much like the word cactus and cacti. A lot of people will say octopi if you see multiple species or multiple individuals. Octopus also works, just like moose and moose. You don't say mooses. So multiple octopus can be octopus. The actual correct term is octopuses. And even that is a translation of what the real term is. So octopuses is correct if you use it. Your friends can't make fun of you. But if you were to say this to them, octopodes. And again, octopodes might be something you've said too. Octopodes is actually the truly correct translation of multiple octopus species. Octopus, octopi, and octopuses are all completely acceptable. This is a weird nerd out moment for those of you that enjoy the English language and how we come to this Latin weird taxonomic hierarchy. A lot of the terms we've translated basically over to the English language come from Spanish, Latin, things like that. Octopus originally stems from Greek language, and there's no direct translation over for the multiples of the species. So the true translation over to English is octopodes, not octopodes. But as far as common zoo vernacular, all of these are acceptable now. So you are welcome to say either. Do not ever feel like you're going to get called out. You always have this to fall back on now if you want to sound like a real fun party guest of your explanation of how octopodes is the true word. But if you ever get called out, fall back on this saving grace and know that there's not a true translation because no one used the word octopodes. If you went to an aquarium and saw multiple species of this and you said, look at the octopodes, you'd get a lot of weird looks. And again, unless you want to launch into this rant, not going to be necessary, but you would still be correct. You'll be correct no matter what you use. So remember that on tests as well. It's an acceptable vernacular no matter what you're talking about. Octopi, octopuses, octopus as a plural. But yes, octopodes is, believe it or not, the correct way to do it. So the term cephalopoda, again, if we're breaking down our root words, we have cepha, meaning head, and apoda, or just poda, meaning foot. I know I harp on this quite a bit, but hopefully it's starting to manifest itself. Getting a hold of these root words is so important for learning strategies just in general. You could be confused still about what animal you're talking about or what you're working on, but if you can pull little tidbits of information, you can start. It's kind of like finding I guess the analogy I use is if you get these root words, it's like finding the border pieces on a puzzle. You might have no idea what the puzzle is going to look. Let's say you lost the box top. Um, but if you can start putting the border together, you start getting a little bit of a picture of what it might turn into. That's what root words do. They allow you to extract little tiny bits of information, but if you do that a couple times, all of a sudden the picture starts to take form. So as far as the current and extinct members of the cephalopod group, we have a huge diversity of fossil species. A lot of them look like what you see on the left side of your screen, because again, that's a very hard shell that presents itself very well through fossil records. The extinct cephalopods in general, so again, animals that are not alive anymore, the extinct cephalopods are known as what we call ammonites. It's the term we basically apply to any prehistoric cephalopod fossil we might find, no matter what it looks like. And to be honest, there's very few fossil records of, let's say, squid, for example, certainly octopus. When we start talking about internal skeletons, you'll see why. But yeah, there's very little fossil records left. But thanks to what we call the ammonites, we do have that kind of origin species. Again, keep in the back of your mind, whenever we discover a fossil, you've got to give, I don't know, X million amount of years, like consideration. It's not like that fossil just showed up. That's the first one we found. So when we go back in time and we say, this is the first time we found this fossil, you know we have millions of years predating that to get that fossil to that point where we even found it. So just keep that in mind if you're doing like personal research, anything like that, and you hear, oh, that was 10 million years longer than my instructor said at the college. Of course it is. Like we're discovering new stuff all the time, redoing carbon dating, but we don't just find fossils. There's millions of years leading up to the finding of that fossil. But here's your original ammonite species. Interestingly enough, and I always think this is cool to tie into the human history side of things, ammonites get their name and history and foundation from the Egyptian god Ammon. It's the ram god, and you can see from the ram's horn coil. This is a weird part. If you kind of are fascinated with this, I always am. Whenever the human explanation for everything comes into play, I always think it's really interesting to find how we as humans have come to understand the natural world originally from a almost fable standpoint, where we have mythology and folklore surrounding these animals, to now, of course, a much more science-based. But I think it's important to tap into the origins of how this animal came to be where it's at. Elephants, for example, if you've taken mammals or maybe seen one just on your own because you're weird like that, ever seen an elephant skull? When they first uncovered elephant skulls, people started to believe in cyclops. If you've ever seen one, the area where the trunk would be and the entire rostrum is just one giant empty cavity. And it looks like a cyclops skull. And for a long time, it confirmed the belief that cyclops once roamed the earth, which if you still believe that, it's cool. I like the imagination. It's very, very cool. But yeah, the idea that all these fossils, before we had the carbon dating and the science, really did have a place in human history, I just always find that fascinating. So enough with the side topics to enjoy. We are on to the class Cephalopoda. You can see just from the picture here, we're going to be talking about a myriad of species. A lot of people probably hone in on the bottom right here to our octopus, but we've got cuttlefish, squid, and then of course nautiloids aren't represented just yet. They will be soon. So within class Cephalopoda, couple huge evolutionary jumps here. The first of which is a closed circulatory system. That is a big deal. It doesn't sound like much, but a huge deal. Again, because of the evolutionary steps we have to take for this course, talking about invertebrates in general, it's weird to see these leaps forward in evolution and then what seems like a step back. But every step of the way, of course, we'll explain why we're studying these animals next and next and next. There's changes every time. But here's a big one. We basically have a very advanced mollusk suddenly developing a closed circulatory system. What this means, as opposed to a lot of your other invertebrates, most of the time you're talking about hemolymph is the invertebrate blood, and then a hemocoel, which is basically a giant cavity that just has invertebrate blood sloshing around in it. Here's kind of our first jump into a closed circulatory system. For those of you at home, that's what you have. A closed circulatory system where we have a heart, veins and arteries. Of course, arteries coming from the heart, veins going back, arteria filling in the in-betweens. But a closed circulatory system is a huge deal. It means we have a brachial heart that's going to supply the gills directly with blood, and a systematic heart that will move blood throughout the rest of the body. Again, when we're moving blood around, I know this is pretty simple anatomy, but just in case we need to catch up, putting blood towards an area or pulling from an area, for example, pulling from the gills, when you're providing the gills with blood, they don't need blood. You need the oxygen that's coming from those gills to come back. Again, as we always talk about with these highly vascularized areas like gills, when blood enters the gills, not only are you able to passively take on that oxygen, you're also able to passively release waste gases and waste within the blood. The systematic heart moves blood through the body. Of course, if we are concentrating blood in one area, whatever that specific organ or muscle is doing is going to increase productivity. So the closed circulatory system here is a big deal. It's a huge evolutionary leap. Of course, after this, we're going to jump back into like chelicerates, which just have, again, that hemocoel filled with arthropod blood sloshing around. But for now, this is a very big step for cephalopod kind. What you would call the foot, again, another big time mollusk trait. The foot itself, and again, this is what you would see if you bought a mystery snail from the pet store. And what's pressed against the glass, that is the foot of the animal. The foot in this case has been modified to form very flexible arms. We'll talk about that shortly, but arms is the right term. It's not tentacles. Everybody wants to say tentacles. I get it. It sounds way cooler. I totally understand. Like if you're in a submarine and get attacked by a giant squid, you don't want to say, oh my gosh, its arms are all over us. You want to say, I've been attacked by a tentacle. It's much cooler, but arms is the correct term. Tentacle needs to be applied in very specific situations. The siphon itself, you'll sometimes see this depending on what you're researching. The siphon can also be described as the funnel, as another term you might hear. The ganglia, these are the neuroprocessing centers that make up the brain. And in this case, we do have an actual brain, which is not unique by any means, obviously on the scale of what you've probably studied. But here, the brain is going to be unique in a myriad of ways, most of which we'll get into later. But it is a brain encased in a cartilaginous. So again, we don't have a calcium-based skeleton yet, but is encased slash semi-protected in a cartilaginous skull or cranium. The lines on the dermis itself function all over the body as sensory organs. So there's it's like a giant neuroprocessing center all along the body, and the entire body is sensitive to both touch, light, uh, predation, avoidance, you name it. The animal is just a big giant detector for everything. Again, light, colors around them. Um, the entire animal is one big sensory organ, which allows them to exhibit a myriad of behaviors. Again, very different from mollusks in general, but they're still there. It's just, again, this is what happens when you start all the way up in a new phylum and then have millions and millions of years to branch off and do your own thing. They're still mollusks, but obviously this is a lot different. With, again, that siphon slash funnel, you're going to have an in current and an X current siphon, in and out. Suck water through here, push it out this way. You can aim this funnel depending on which direction you want to move. But it's a very unique way to come up with a locomotory system of, and we'll talk about this much later on. Anytime, though, we see an animal that's using its environment in order to manipulate how they move, that's so efficient. It'd be like us, like I know right now we have electric cars. It's a good step. But imagine if your car just drove off sunshine and wishes. Um, that's the idea. You're using the naturally occurring environment in order to transport yourself. So they're just using ocean water. They don't need really strong muscles, which they do have. But they don't need strong sprinter legs, things like that. They just need the ocean, which, believe it or not, is around them constantly. They're able to move and become so efficient because they're using their own environment. The funnel here can be manipulated, again, direction-wise. It's also going to be manipulated direction-wise because this is where they'll expel ink from. So for predator avoidance, these animals can expel ink and direct that ink at whatever unstoppable force is moving towards them. The rest of the party comes here with the ctenidium and the radula. These are going to be present in all cephalopod species. Couple things of note here. This is arguably the most important point as to why we've gone back and studied these animals and now understand that they are indeed mollusks. The ctenidium is the comb-like respiratory organ, or what we call a mollusk gill. It's comb slash feather-like. Basically, kind of picture spirit fingers going on and water moving past that, and then do double spirit fingers like the dove flying. It's a feather or comb-like respiratory organ, but it's present in every single mollusk. Again, these animals are far and away more advanced with the stuff we're going to talk about. But again, at the end of their day, still mollusk species. The radula, this is the other across the board mollusk characteristic that we're going to find. It's a rasp-like structure. The closest equivalent for your comprehension would be just a tongue. For the example we're going to use today, picture a cat's tongue. If you've ever been licked by a cat, you feel how abrasive it is, and that's intentional. The cat species as a whole, the tongue is designed actually to remove hair from whatever prey they've taken down. It rips off hair so you can get to the meat without coughing up the proverbial hairball. The radula is a lot like that. It's not necessarily a tongue. It's a scraper, but I don't know. Another example, I guess, would be like maybe a zester, or a cheese grater type thing, where it's a tongue-like protrusion that chips away at whatever prey item you're consuming. The important part about that is the reason we have that zesting appearance, so that very fine micro grating, is that's the only time you have to actually masticate or chew food. So very important that the radula is there because that's the only way to break down what's entering the body. That's our first chance, first and only chance to quote unquote chew. We have a beak that pierces prey, and then the radula that comes in and micro shreds it for processing into the rest of the body. So ctenidium and radula are the important aspects that tie these animals in with the rest of mollusca. Again, from the visual here, I'll give you a second on this slide. You can see the radula, see the ctenidia. Not seen it yet. There's radula, ctenidia. Two very different looking animals here. The visual aspect makes it confusing, but shouldn't. It really is the same organism. This is just the same analogy as we talk about with sharks, skates, and rays. Sharks look nothing like rays, but rays are literally just pancake versions of sharks. And same thing here. We've got an octopus versus what we know now as ammonites, but nautilids, and such a different visual representation, still looking at the same animal. The same parts are going to be there. So again, head foot. This is going to lead to some interesting conversation. The shell, if it is present, is going to be divided by septa. A lot of the times, these animals won't have a shell. It's going to be very reduced, lost in some species, most notably octopus, but certainly slimmed down. Nautilids, obviously, again, are the outlier with this giant externalized shell. If the shell is externalized and present, it's going to be divided by what we call septa. Septa are basically calcium carbonate secretions left behind as the animal moves forward. But because that shell still exists, there's always going to be a connective strand of porous calcium carbonate tissue that spreads all the way back to that origin part of the shell. So again, back to that idea of a coil here. Even if the animal sits here, there's always going to be a connection back to here through this strand of tissue. As far as the variants in shells, we obviously have 4 species here, so there's 4 kinds of shell. The nautilus is the easiest because obviously they have this huge externalized, very visible shell. It's multi-chambered, gas-filled depending on buoyancy needs. The cuttlefish with the internal shell of the cuttlebone. You may, if you've kept birds or worked with birds, heard of cuttlebone before. A lot of the times now, cuttlebone is manufactured or synthesized, but we did used to get a lot of our cuttlebone from cuttlefish themselves. It's and again, if you've worked birds, you've seen them. Looks a lot like what we see right here. This is also very analogous to what we're going to talk about with the squid as well. But a hard piece of calcium byproduct, basically. Again, these animals are invertebrates. We don't want to say it's a skeleton, but this is as close as we have to a skeleton. It's just their shell. In this case, will be internalized. So cuttlebone in squid, it's called the pen. I'm not sure if they still do it. If you've had the chance to dissect a squid, maybe in junior high or high school, things like that, you've more than likely seen the pen. When I've done dissections, that's one of our kind of awesome, wow, that's cool points. When we cut into the animal, go through the whole dissection process, and then removing the pen is always kind of one of those cool aha moments. Octopus have no shell. Of course, there's got to be an exception to that. We can't just say no ever. There are a couple species with a vestigial shell. The reason this is important, the reason we're talking about this, is A, might show up on a test, but B, the idea of having a shell is a pretty big mollusk thing. And again, aside from the ctenidium and radula, octopus begin life with the vestigial shell. So in embryonic development, they have an internalized shell. They just use it and absorb it as they mature into adulthood. So this is one of those parts where I'll apologize. When we have a huge grouping of animals and then step in and start separating them to organize them taxonomically, it's always nice to split the group 50-50. Not the case here. In this case, we're going to take all the nautilids out, and then leave everything else in the other group. It does make a lot more sense here than you'll find in some other invertebrate species, though. Separating out the nautiloids. Nautiloids are very unique in both their appearance, their behavior, the way they locomote, and things like that. The coleoids are going to be everything else: squid, cuttlefish, and octopodes. There, we used it. Um, the coleoids make up everything else. And while, again, that's a little frustrating from a science studying standpoint, it does make a lot of sense when you get to these animals. Nautiloids, again, very unique outliers, and the coleoids should be studied differently. They just should be. There's justification for it, and it does make sense in this scenario. With these animals, we're going to harp here for a minute because this is probably the most confusing part for a lot of people. We have three very distinguished regions. First, the brachial crown, the head, and the mantle. And the reason, I'm gonna try a drawing here, everyone, so bear with me. The reason this is so important is if you tell a lot of people, where's the head of an octopus? They're going to tell you one thing, and when you say, where's the body? They're going to say something else. So here comes my amazing drawing. One, two, three, four, 6, 7, 8. Now we've done them justice. Now it looks like a real octopus. Here we're going to have eyes. Maybe a fin or two. This guy's looking cool. I'm going to sign this one because it's a beauty. That Bob Ross would be happy with this. So let's talk about the distinguished regions here. Brachial crown. I'm going to change the color of my pencil real quick here. So brachial crown is just the arms and tentacles. So it's everything here. Here and down. That is our brachial crown. It's the arms and or tentacles if they are present. The head is going to have the lateral, very prominent eyes, but the head's really this part. It's this little cross section right in the middle. It's also going to contain the ganglia, so the brain. And then the mantle is everything up from here. This is important for a couple of reasons. One, most people couldn't tell you where the head versus the body was. A lot of people think this is the head, everything down here, this is the body up here. Not the case. The reason this is so important is the brachial crown is what was the foot. So I want you to take a second here. We know what cephalopod means, right? Head, foot. We know that here's the head now. And we know that the body's up here. Take a second and just understand the evolutionary process that occurs here. It's weird to even say out loud. When we're talking about an animal like a cephalopod, they are named aptly so. This is literally a modified version of what you've seen stuck on the tank from that mystery snail. And imagine the evolutionary process that has to occur for an animal to go, foot's the most important, then my head, then my body. If hopefully at home you're all picturing a human being looking like that, you understand the crazy adaptations these animals have had to undergo in order to become like this. You go foot, head, then your body on top of you. I don't think they're going to get too many swipes on dating apps if that's truly how you look. The foot comes first, then the head, and then the mantle, which is going to contain all the viscera. Again, fins if present, but all the guts go up here. They sit right above your brain. You've got guts on top of your brain and your foot right underneath your chin, basically. Crazy, strange adaptations. But again, this is what millions of years of evolution gets you. And I don't think at the end of this lecture, anyone's going to argue with the amazing intelligence and fascination these animals draw. It's not a bad model. It does seem very weird when we break it down, but that's where head foot comes from. And yeah, there's nothing wrong with this model. I'm a big fan, but it is very counterintuitive as to how nature would design a creature. With nautiloids, despite we had that amazing drawing earlier, that idea of the iconic kind of 8 arms being present, nautiloids have around 60 to 90 arms. Sometimes they're called tentacles, but more often than not, they're referred to as cirri or cirri. These are much more finger-like projections. Again, back to that spirit finger analogy. Now picture you have 90 of those going. It's very delicate movements, all very capable of performing some finite motion. So they're very dexterous, but much smaller in scale. With all the coleoids, again, this is cuttlefish, squid, and octopi, they're going to have that single ring, again, that brachial crown with either 8 or 10 appendages. Usually, and again, I'm saying usually, not a guaranteed rule, but usually across the board, if you have 8, they're all arms. If you have 10, we're looking at the addition of what we call tentacles. There are, of course, exceptions. We'll talk about a few of them, but usually if there's just 8, they're all arms. With nautilids, again, we talked about this just a second ago. If there is a shell, the animal is going to keep growing, but only inhabit the last section of that chamber. Every time the animal moves forward, it's going to secrete behind it what we call a septum. And again, multiple, we're getting septa. These are the chambers in the shell that start to grow as the animal secretes calcium behind it. But again, this is another odd thing. When we think of, again, other mollusks, let's say snails and things like that, we always think of the animal as being able to retreat inside the shell. Not the case here. The animal is really going to inhabit that last section of the septum, secreted behind it. It's not able to retreat back inside of that shell. Again, as we talked about, this is a better visual representation of it. We have a very unique bow plan here, the initial body plan of the animal. We have the animal starting off very tiny down here. As it grows, again, it's going to secrete a nacreous, nacre is the thing that makes up pearl, nacre / calcium carbonate structure behind it, and it keeps moving forward. Again, it is not able to retreat inside this shell. It inhabits only that last chamber. But in order to make that shell relevant, it's going to keep this magical structure. It's arguably my favorite term in zoology ever, the siphuncle. It is very fascinating how it is utilized. The siphuncle itself is going to run a lot of the processes these animals need for survival. So again, every one of these is as the animal grows. I don't know if there's a old tale of how you can tell how old the nautilid is by how many chambers it has, like rings on a tree, but we certainly can see the progression of the animal as it grows. So to the point of the siphuncle, it's a calcareous tube, but it contains living tissue. So very veinous and a little bit of muscle, but mostly blood flow through this living tissue. It connects all the way back to that origin little piece of shell where the animal started. It's going to run throughout the shell of the animal. And right here, I'm going to leave this on the screen for a second so you can process and digest it. It's going to help the animal adjust buoyancy in the water flow, in the water column. Let's think about it for a second. If you're an animal that lives only in that last little chamber, you don't have the super long arms to move about the bottom. How are you going to adjust your buoyancy in the water column? So what do you guys think? Like, how are you going to adjust buoyancy in the water despite this very heavy shell on you?
So the process is actually really cool, I guess. Like they control it, but it's really instantaneous. And what kind of happens is so like through osmosis they're going to control gas flowing into the chambers as they're like changing in salinity, So changing the siphuncle into higher salinity will allow the, I guess, like diluted chambers, diluted water or fluid, if you will, to move through the siphuncle, and then gas will be able to move into the chambers, which is going to allow them to control buoyancy. And they're not like going up into, like the surface. And it's not really like they're pumping gas into their chambers like we would assume, like, oh, they're just, pushing gas through there. Like the energy flow or the energy absorbed is actually like the energy absorbed. The energy being used is actually to absorb the water through the osmosis. The gas moving into the chamber is just kind of like a passive process through that. And it's all happening instantaneously, even though there's multiple little things going into it. And then they're able to control buoyancy just enough, like as the chambers are varying in size, they can kind of control where they're moving and be able to move, whether it's an inch or a couple inches through the water chamber, to kind of adjust themselves again, like instantly, and just kind of almost like flow through the water there.
Perfect. And I really want to touch on a point that was made here, was the term passive. And that is the key term to use when describing this process. Um, the entire process, just like we talked about earlier with gills, whenever you're transferring oxygen in through a vascularized area, you're also able to passively release waste gases, bringing the good stuff in and releasing the bad stuff through just the meeting of the two, a meeting of the minds. And yeah, passive is the word I really want to focus in on. And again, these animals, hands down, compared to an octopus, octopus are exciting. The intelligence levels out of the roof, like incredible. But again, a big thing I like to harp on with invertebrates is the simplicity of the model. These animals, that huge shell would be very cumbersome and a huge burden to most animals. These animals, through that passive exchange of osmosis, are able to adjust their buoyancy in the water column. So again, let's go back to the gills. As these animals float along seemingly mindlessly, very simple, oh cool, you're an animal with a big shell. If you remember osmosis, oops, let me change the color of that, I apologize. If you remember osmosis, water wants to travel from low to high salinity to achieve a balance in nature. When you do this osmosis flow, we're exchanging gas and taking on oxygen, releasing oxygen or waste gas. A lot of things are occurring here, not just the movement of water. And so, again, let's think about it this way. If these animals wanted to float, I think your average person would say they fill up with air. Well, there's not a ton of air just floating along in the ocean. The most logical way would go up to the surface and take a breath. But of course, with this giant shell, you're never going to make it to the surface to take a breath. So, yes, a simple animal, but they're able to change their salinity of their blood on a whim. They can say, oh, I need to go higher. I'll change my salinity. And I'll either absorb water into my bloodstream, flush it out my system, or I will passively release waste gas into my shell and then become more and more buoyant as we move through. So there's a lot going on, much more so than just sucking up air and hoping for the best. Again, simple, when it's been around for hundreds of millions of years, simple works, people. It's a beautiful design by a very, very simple animal. Oddly enough, one of the things that maybe became oversimplified does seem kind of silly in today's age. They've lost what's called the apticus. And this is the plate, again, that you would see on the mystery snail, if we're using them as an example. That ability to close and retreat. Prehistoric and extinct nautilus used to have this. And for whatever reason, I mean, again, I can't argue with hundreds of millions of years of evolution. They know what they're doing. It does seem odd to me that they would leave this off. The apticus is basically the lid that would stick out here and then snap shut if the animal was ever under threat of attack or things like that. For whatever reason, and again, I'm not going to argue with them. Bravo, brave nautilus. You've done a good job for a long time. but they're going to lack that apticus to actually close the shell off. Again, the term extant here has been used a couple times. Again, remember that's just not extinct. It's currently living today. All the other extant cephalopods, other than nautilid, again, with the cirri, have 8 arms. As we said before, if they have additional arms, those are now either filaments or tentacles. And as we'll get to later, there is some debate, especially once we talk about the vampire squid. A little debate out there, but just know if there's over 8, the rest are usually tentacles. Again, asterisk on the vampire squid, because there's always got to be an outlier, right? That's what makes this field fun. As far as size differences here, I don't know if we're going to find another species where the size difference is so astronomical. We've got our giant squid, and I know some of you at home are screaming at your screen and saying, there's the colossal squid too. Yes, there is. Giant squid are longer though. With giant squid, and again, I don't want to burst anybody's bubble, but a lot of the times those animals are usually decayed and bloated when they wash up on shore. A lot of the pictures you can Google on the internet are kind of forced perspective, kind of an animal that's gelatinous and bloated. So of course, sizes are going to be exaggerated, but that shouldn't downplay how big these animals are. You're talking about 60 feet long, and that again is the mantle plus the very long arms and then extra long tentacles that we'll find. You're talking up to the 700-pound range, very, very large. And then on the flip side of that, we've got a myriad of octopus species that rarely get above about 1 to 2 inches, the smallest of which being way under half an inch. These pictures don't do it justice. If you've had the opportunity to go to a museum and see the recreation, or in some cases, if you've been to DC or places like that, see an actual preserved specimen, they are kind of humblingly large, very, very huge on this side, and very small and adorable on this side. This picture has clearly been enlarged for viewing. Really, this animal's about this big. That little middle part of the E there. So adorable, very cute. Everybody likes small, cute animals, and cephs are no exception. But that disparity, again, is what millions of years gets you as far as adaptive radiation. All right, we've seen the cute stuff. Let's get into some science. That's where we start talking physical characteristics. The behaviors of these animals, as we'll talk about throughout today, are astounding in how many cool behaviors they're able to do, from both their intelligence, their behavior, ways you can train them, the husbandry for them. But a lot of that starts right here. We've got a couple things occurring here. We've got the presence of various cells that are going to do their respective works. The chromatophores are basically what we consider color changing or light reflecting. Again, guys, because understand how light's going to reflect. And then iridophores, which are iridescent pigments, which confuses people a lot. Can anybody tell me the difference between a chromatophore and an iridophore, and maybe their roles in how cephs are able to manipulate their appearance?
Yeah, so like you said, with light it plays a huge difference on what pigment is being displayed. So firstly, the difference between chromatophores and iridophores chromatophores are organs within the skin made up of different layers of cells. Iridophores are actually one of the layers of cells. It's just basically iridophores are stacked cells. So I like to think of, those plastic clipboards that you can open, keep your papers in. I think of them like that, and the way that they angle and move. Uh, th- Sorry, give me 1 second. So iridophores don't contain any pigment, but chromatophores do contain pigmented granules in various regions of the organ. And the iridophores actually play a huge role on what color is being displayed, as I've said, because the iridophores allow or reflect light at different and really kind of make it dependent on what color is being seen, what color the light is being reflected and how far away and at what angle. So within the chromatophore, there are, like I said, pigmented granules, and the chromatophore is more quickly responsive to neurological impulses versus the iridophores, because the iridophores do reside inside of the chromatophores in a lot of these species that we've been talking about. And so they're almost activated immediately, instantly through neurological impulses, whereas the iridophores are kind of a secondary reaction. So their reaction is a little bit more slower because they are functioning within the chromatophore. So of course they get the secondary kind of response. They're reacting to what the chromatophore's function is, depending on that response. So there are different granules, each one in a different area of the chromatophore, and depending on the response, the chromatophores expand or contract the regions of the granules, like if you want to call them like cells full of granules or sacs full of granules, because that's basically what it is just an organ that contains sacs. And so when the impulse comes through from uh, the octopus, oh no, for example, not to anthropomorphize, but oh no, I'm in danger. They either, their impulses will go right to they'll trigger the chromatophore, right? And then the chromatophore, depending on the situation or where the surroundings are, the coloration of the surroundings, they're able to expand, contract, utilize different parts of the cells within the chromatophore to show XYZ colors like yellow, red, brown, black, white. And so the important part here with the light is they reflect white light off of these different color or pigmented granules. And also the iridophores, because they're iridescent and highly reflective, they allow the variation of the color you're seeing. So like a lighter color, darker color, how intense, even down to varying from distance. I believe I've read that, they're unsure, but I'm pretty sure I believe, based on what sorry, I'm just rambling now. Based on what I've read in articles and stuff, a lot of scientists do believe that it's uh, also dependent on distance, how far away an organism is from seeing this animal, how is dependent on the color that they're seeing. But yeah, it's not even in iridophores. It's not even color at all. There's no pigmentation in iridophores. There's just reflection of light, and again, that does play a huge role, working together within the chromatophore on what color you're seeing.
Okay, first off, the number one point there was that none of this matters without light. Excellent information. It's exactly correct with how these animals work, but you need light to begin with. And I think that goes so unnoticed when we think about the amazingness that these animals exhibit as far as color change, as far as behavioral adaptations. You need light to get that whole process started. And as was talked about iridophores, the reflection of light depends on light being present. So, of course, the deeper we go, we're going to have to come up with new adaptations and things like that. But a lot of the amazing color changing behavior we'll see, we'll talk about that in a little bit as far as the different colorations these animals can exhibit. You need the presence of light. To put into perspective, the way I always like to think down here with iridophores is picture them like a disco ball. A disco ball on its own, obviously very cool, but does literally nothing aside from look cool. does literally nothing, though, without light shining on it. They're able to reflect light, but light has to be present. Otherwise, it's just a hanging, sad disco ball. Chromatophores, again, as was explained earlier, the pigment-containing ones are going to reflect different light from them. And hopefully we don't have to dive too far into this. I know you guys understand how colors work and prisms and things like that. But when an animal changes color, you're not seeing what color it changes. You're seeing what light has refracted or bounced off that animal, if that makes sense. The other unknown here, the other variable that we're not taking into consideration is how these animals see it. We only see what we see. And I know that's a very stupid and easy saying, but we're only seeing what we're seeing. That's how our brains are manifesting and figuring out these colors. The actual colors that are showing may appear different if other animals are seen in different chromatic lenses. Same with the iridophores, with the idea that maybe this light reflection is like instead of the just glare, for example, off a mirror, maybe it is a dazzling array or a certain sparkle or a glitter confetti monster rainbow. Like we don't know what these animals are interpreting because we don't see in that specific lens. So we've got reflection of light and then reflection of color through light, both needing light though. The reason we're talking about this and to put it in perspective is once we get in deeper waters, this has to switch gears because changing colors no longer matters. Iridophores, completely irrelevant now because there's no light, unless, of course, you're able to make some on your own. But yeah, a lot of science here, a lot of amazing things, but they're only amazing for the way we interpret them. It's difficult to know how amazing they are because we can't see in the lens of let's say, a male and female octopus or rival male octopus competing with one another. The colors they're expressing might seem one way to us, but different color spectrums means different things to different animals. And then again, none of this is relevant without the presence of light to begin with. So this is definitely not test worthy, but it is worth note. We've got a myriad of colors that are being now studied as far as their hue that they exhibit to us under white light. The colorations that come into play are becoming more and more important to the biomedical human field. Melanophores being the main one. This is the study of pigmentation in humans with as far as either sun exposure, locality of the equator, things like that. Not necessarily a racial study, much more on a cellular level of what these cells are doing, what are they manipulating themselves for in order to, again, not color, reflect color back that way. So there's a ton of relevant studies here. I mean, you could go into the field of invertebrates and then spend another couple of years specializing in cephalopods, and then another couple of years getting to the genetic level of understanding what each of these fours basically do. Very fascinating stuff with them. To add on to all that, that's when we come up with photophores, because why not add something else to the fray? With photophores, again, as was explained earlier when we asked the question, the presence of light is always very relevant to how either iridophores or chromatophores are going to play themselves out. Once we get into deeper bodies of water, and again, when I say deeper bodies, think about how quickly light dissipates in water. Picture the deep end of a pool, how quickly you lose light. Now picture going 100 feet down or 200 feet down. Very quickly, you lose that presence of light to make any of these things relevant. So of course, cephalopods have come up with something unique to occupy that niche. And that's where we get the development of photophores. We have two fundamental types here, intrinsic and bacterial photophores. Intrinsic is unique. We're going to talk about that second. Bacterial photophores are much more common. This is hosting a photogenic bacteria. That does not mean it takes good pictures. It's a bacteria growth that basically lives in the ink sac of the animal, and in conjunction with protein strands and the ink the animal releases, basically results in a light phenomena. Now on to intrinsic photophores. This is a biochemical reaction between various proteins in the animal. It's not rare, but it is rarer than bacterial photophores. The animal's literally able to combine parts of the body, chemicals, protein strains, things like that, and produce light. Now, unlike the, let's say the anglerfish, I think is one everybody's familiar with, where there's a glowing angle, literally a prey attractor dangling out there. A lot of the times, your cephs don't necessarily need that. They're very crafty. They can move around pretty well. They don't need to just sit a lure out there and wait for something to bite the end. There are a few occasions where an animal will have some what appears to be bioluminescence, but usually not the case. What it's used for primarily is defense. Again, as you can see at the bottom of your screen here, associated with the ink sac. There is a reason for that. Again, let's think about the absence of light now. Think about being towards the surface. If a predator comes near you, you're going to ink. Picture Finding Nemo where, oh, I inked myself. Ink is going to disorient, distract the prey, possibly cause some damage and harm. But when you're in the absence of sunlight, all of a sudden ink doesn't really hold up. Instead, we've got to go the complete opposite direction, where now instead of ink, we need a flash bomb grenade. And that's what these bacterial photophores do. It's bacteria that basically lives, think of it almost like gut microbes, live in that ink sac. And when the animal expels ink, instead of just darkness, because you're already in the dark, now we've got a confetti, glitter, rainbow sparkle, something from My Little Pony, huge explosion of like a firework going off. That's much more disorienting in a absent of light environment than ink would be. So now we've gone the opposite direction, where instead of inking to disorient, now we've got explosions of light to disorient. And so the way these photophores work with the animal are very unique. One of the things I do want to point out here is this idea that all these animals bioluminesce, so put off their own light. Yes, there are plenty of animals that do that. I'm not trying to narc or harp on them. Most of the time when you're seeing those animals, though, it's from the view of a submarine with light shining on them. Very rarely do we have manned or unmanned submarines go down with the lights off and hope to see something. So just keep that in the back of your mind whenever you watch videos, nature documentaries, and things like that. I love deep sea animals. I think it's arguably one of the coolest things out there. But a lot of the times, the way you see them under the light of, say, a submarine isn't necessarily how they're presenting themselves in the absence of light. It's very rare to get a fully in the dark shot of an animal that deep sea that naturally presents itself that way. It's the presence of light that's changing how we're interpreting it. So again, cephalopods, totally cool. Nautiloids sit outside of them, though. The coleoids are very unique because nautiloids lack all these things. Nautiloids are going to lack the ability to ink. Again, you got to figure the area that the animal occupies is at that terminal end of the chamber. They're also going to lack especially the CNS, the central nervous systems in nautilids. Again, living at the back of the chamber, that comes into play. Image forming with the eye. Nautilids, very minimal eye. It's not these huge, prominent lateral eyes. And so while we've talked about nautilids, we've also kind of crossed them off our list so we can move forward in the rest of our study of cephalopods. But still, again, I don't want it to seem like I'm harping on nautilids here. Amazing animals for what they're able to do. The octopus species and the rest of the cephs, the coleoids in general, cannot have that claim to fame of being able to, as answered earlier, change the salinity of their blood on a whim just in order to stay buoyant. So now that we've moved on from nautilids, again, nautilids able to do what seems like less, but on the flip side, they're able to live a lot longer. We're moving on to the coleoids. Here we have an animal that fascinates, or a group of animals that fascinates us with their intelligence, their behaviors, things like that. But you've got about 4 or 5 years to live at best. Typically, the colder water species tend to live longer. The warmer it gets, so the smaller octopus species that would live in, say, a coral reef, they're basically the equivalent of like a mayfly. You've got about maybe 4 months to live from birth to breeding, hopefully, and then dying off. 4 to 6 months is about average for them. And then your larger octopus species also have very short lifespans. GPO is referred to a lot. Don't get it confused with GPP, the gross primary productivity that we talked about. Your GPO are your giant Pacific octopus, most notably the North Giant Pacific octopus. Probably, and if somebody calls me on it, I'll concede. I'm pretty sure, though, it's the most represented octopus species in all the aquaria you work with. Depending on where you've been, aquarium-wise, I would guarantee any larger aquarium, not like accessory animal aquarium, would more than likely be exhibiting Giant Pacific. They are so, I'll just say it out loud right now, people, magical. The way they interact with their surroundings, with guests on occasions, the way they usually just sit in the tank is super fascinating. They could not even be moving, and people are like, wow, that's incredible. Very amazing animal to observe, certainly work with if you have the opportunity. This should be a life goal for you to work with one of these animals. But you're limited to a couple of years of interaction through no fault of your own, so you don't have to feel bad about this one. This is the life cycle for most of our octopus species. And that brings us to the majestic, the great, the wonderful octopus. So, pretty decently sized group here. Again, this is just off what we know. As we mentioned earlier, the tropical waters seem to be smaller, and the colder, and again, I want you to keep in the back of your mind, it's not just colder, it's the deeper we go. There's no coincidence that the largest cephs live significantly deeper than everything else. The colder we go, the larger we go. There are no freshwater species. They're strictly marine. And they're going to be, for the most part, benthic. Again, remember, benthic is no matter what level, going to be coasting along the bottom. There's always exceptions. And to be honest, the exceptions in this case, we don't know a lot about. Animals like the giant squid, like the vampire squid. We know so little about these animals. Some of them do seem like they're free swimming. Giant squid we know almost nothing about, to be honest, to confirm anything scientific-wise. And so we know there's asterisks, but for the most part, the animals that we do study, which again, if we think of the layers of the ocean, we're talking the sunlight zone. So here's the body of water that is our Earth's ocean. We know about this part. It's not much to base entire species off of, right? That's probably not a fair assessment of everything down here. But that is where we're at right now. That's what we have to work with. They are completely solitary animals, except when it comes to mating. Mating is going to be a very interesting subject as well, because that's what the world of invertebrates does for us. But you got an animal that lives its entire life alone, almost from the time of birth. We'll talk about that in a sec with how the eggs are hatched. But a really solitary animal that's able to do so much with no coaching or parenting. Again, now that we're off the nautiloids and onto the coleoids, we've got entirely soft bodies for the most part now. And with most of our octopus species, there's not going to be a internal skeleton. You've basically got the beak. That's about it. With octopus, as opposed to squids or cuttlefish, they're not going to have fins either, with, of course, a few exceptions. There are a few species out there that seem to be, and again, just from a appearance perspective, not a science perspective, but seem to be like the almost missing link between squid and octopi, where there are some suborders that have fins and an internal shell like a cuttlebone or a pen. So we do have that transitory animal that gives us that confirmation of the divergence of the species. This is it right here. This is the only really solid part on the animal's body. Brings up a couple challenges. That means if the beak can fit through something, the rest of the animal can too. That makes it extremely hard for aquaria keepers. We'll talk about that. It makes it hard for figuring out what they're going to be able to do, even housing them in the first place, much less keeping them housed. And I'm sure by this time you guys have all seen a video at some point of octopus doing cool things. I remember a couple of years ago there was one of an octopus that had crawled onto I don't remember what kind of boat it was. I don't remember if they were catching crabs or fish or things like that. But it literally squeezed out through like the air duct and went out the side of the boat. Anything the beak can fit through, the rest of the animal can too. Again, yes, we've got a ganglia or a brain, but it's surrounded by cartilage. That's the every time I say primitive, it always makes me laugh because primitive works, people. The primitive advantage of having cartilage instead of a skeleton is that you can fit through anything. It works very, very well. Here we see the mouth itself, the beak. The only limitation here is that the fact that the beak really just does nothing but pierce. A couple things to note here that this is just a good visual representation for it. We'll see the text on the next slide. The beak does the piercing, the salivary gland and the radula do the rest. Salivary gland is going to be very, very important here, but the beak does nothing but actually pierce. And one of the things we'll talk about later when we talk about arms of these animals and how they understand things is this is maybe the only design flaw of the octopus. And that is the fact that the mouth is on the underside of the animal. So you're never going to see what you're consuming necessarily. You have to kind of figure it out. But they have ways around that too. Again, millions of years head start. That's what this will get you. So with this feeding ecology, oops, excuse me, sorry, clicked one too far. Again, that brachial crown and that buccal mass allow them to eat almost anything. I think a lot of people think that they're scavengers that maybe just eat bloated corpses on the seafloor. They are arguably one of the most efficient predators on Earth. Extremely varied diet from animals that can move to sessile organisms, incapacitating crabs, perfectly capable of eating fish that they can catch, snails, turtle, like you name it. Huge variance in diet here. They're going to catch their prey, of course, with the arms. But again, the beak is what's doing the work, and the arms are assisting in that. Again, the only design flaw is that the mouth is on the underside of the animal. They can never visualize what they're doing, and that's where the venom comes into play. The radula is going to be used, like I said earlier, to actually scrape the food. That's kind of the first step in chewing, quote unquote. But we've also got something else going on. And again, it's because the animal cannot see what it's consuming or incapacitating. It's seen it as it approached and during the possible battle that ensued. But as soon as it goes underneath the animal, we kind of lose visual contact. So we need the beak to do a lot more than just hope for the best. The venom that is used is usually prey specific, right? Because that makes sense as far as an evolution standpoint. We're not just going to have mega venom associated with us. We have venom associated with what prey we primarily consume. Usually it's incapacitating. It's not used to actually kill off the animal. It's like a taser, if you will. It's designed to hold the animal still while the radula gets in there and gets to work. So we have arms that are very dexterous and talented, able to bring the prey item in. Then the beak, all it does is pierce, and then it holds on. The beak pierces that salivary gland that we saw on the last slide. Let me go back one. The salivary gland here is used to, of course, coat food as it comes in, but you're already in an aqueous environment. We don't need that quote unquote food lube, if you will. The salivary papilla here, though, this is what's releasing venom into the animal. So let's say we got Mr. and Mrs. Crab out here. Do some cool claws. This one's huge because this animal's awesome. We've pierced the shell, crunch. And then the salivary glands are going to release toxins into the animal to hold it still. The beak doesn't do much other than pierce. Certainly you can move along, but then the radula comes into play and starts going inside the animal and scraping food out and then back into the digestive tract. So piece by piece, this animal is literally incapacitated and then picked apart or plucked apart as the radula does its work. Of course, there's exceptions, one of which we'll talk about in a second here with the vampire squid. Vampire squid's a special creature. This is one of these that has a super cool name. And again, aside from the giant squids and the colossal squid that live deep-bodied, an animal that unfortunately disappoints as far as size, not at all as far as behavior. Like, it's as built, it fits up to the billing, just not size. Excuse me. When you see the term vampire squid of hell, you think, oh my, this animal's pretty BA. We got something going on here. Unfortunately, it's pretty small. Usually 9 to 10 inches is what we're looking at. And I assume at this point you're questioning yourself of what's going on here. You've got an animal that has this terrifying name. It's relatively small now that we know that. It inhabits the midnight zone, so an area completely devoid of sunlight, but does have a unique appearance, and we'll see that in just a second. Extremely underdeveloped chromatophores. Again, as was mentioned earlier when we asked the question, chromatophores, of course they have underdeveloped. Let's think about the history of all animals that live in an area devoid of sunlight. We almost lose that pigmentation. You start to get maybe opaque, maybe even see-through, very bland in coloration. So these animals, because they're still cephs, have that tie to chromatophores, but instead are going to use intrinsic photophores to light themselves as they go. So here's the cute version. Now we're going to go one step ahead. A little less cute, depending on what you think is adorable. But we're starting to see a very strange characteristic here. We're already seeing this kind of webbing between the arms and obviously two very discernible fins. This is where things get interesting. Here's why it actually gets its name. There's your vampire squid. Much like there's cute, there's meh, and oh boy, I understand now where we get that name from. So here's the transition of possibly cute, okay, becoming a little weird. Now things are getting weird. Okay, we've entered the twilight zone. And you've actually entered the midnight zone of the ocean. Up next in this video will be linked after this discussion gets posted. The study and observation of the vampire squid has led to a bunch of amazing conclusions. Can anybody sum up what this video kind of contains and what it goes over?
So in the video, it's the Monterey Bay Aquarium Research Institute ended up doing a study, and they mentioned that it's actually over about 25 years. They study around 200 individuals. And basically what they find is, as you could see in like the pictures, they don't have those typical like really long prey capturing tentacles. They actually have two little filaments, and they will kind of, like in the second picture, I guess, open themselves up and let a filament out and just kind of slowly float. And what they have found is that they'll pull the filament back in and kind of collect everything that's been able to stick to it, and that they're actually consuming marine snow. And because they're in such a low oxygen area of the ocean, they kind of need to adapt to that. And being that they've kind of lowered their metabolism and they have found a way to use what's abundant down there, like prey's minimum, but you have everything falling from above you. So they basically just throw a filament out and kind of float. They haven't really developed a lot of muscle for locomotion, so they really do just kind of collect all of this marine snow and just pull the filament back in and pull it off of there.
Wow, like a ton to process there. All right, let's start with one of the key terms that was used, and I want you guys to remember this one because this will definitely come up later. Marine snow. Sorry for my atrocious handwriting on the computer. Marine snow. This is the closest analogy I have is if you had a fish tank before and you feed your fish and they chomp up all that flaky fish food, it's the little bit that falls out of their mouth and starts drifting down and gets into your substrate. Now picture that through hundreds and thousands of feet of ocean, we've got all this very small detritus falling down. And it does look like marine snow. It's extremely small. And I guess the main point here is if it is, in fact, marine snow and detritus falling down, what makes the cephalopod unique?
That would make them a passive detrivore rather than hunting for live prey. They would be eating basically remnants of what was at one point live, so is no longer living.
Yes, that's like we've got this cool animal already, but that's the biggest breakthrough here. We've got and again, I'm saying world's first now. In a couple of years, hopefully, after you all graduate and go off and study the oceans, I can say there's more. But we found our first passive detritivore. Like, it's an animal that's consuming marine aggregate and particulate, and it's a filter. It's a giant filtration system. As was mentioned, they move along with this filament. I will say now that there's a little debate right now as to whether or not, and I can say from my perspective, they are filaments. But there's argument between whether those filaments should be tentacles, which would make this animal a squid, versus if they are just filaments, which basically are mucosal strands, as was mentioned. If they're just mucosal strands, there's a little bit of argument to make them an octopus. But they've got fins, they've got an internalized shell. Squid does seem like the right housing for them with the filament. The filament narration on the story. And they're filaments. They're mucosal strands that hang out there and collect this marine snow, this aggregate. But yeah, the fact that we found our first very passive detritivore that passively swims along and consumes all this marine snow, it again kind of shatters the mold on everything we just got done talking about how fantastic octopus are at hunting and how effective they are in incapacitating and acquiring prey. Then of course we have one that lives in the bottom of the ocean and glows a little bit and eats all the marine snow. It only makes sense for the animal kingdom to throw us a very cool but very small curveball like this animal. To expand though, on the flip side now, we've talked the detritivore version where we've got these detritivous species consuming literally what everything else has eaten already, and then cleaning up the mess before it falls to the bottom of the ocean. And again, hopefully this all makes sense as to why an animal would operate that way. Think about the nitrogen cycle. If we don't have animals like the vampire squid plucking these particulates out, eventually all that stuff settles, and we just have this buildup of previously, it's just like a fish tank again, overfeeding, where all the food sinks to the bottom, and then we've got this huge nitrogen spike. We need animals like this plucking organic particulates from the water column as it moves down. So hats off to you, vampire squid. On to, we've found out the outlier, even within octopus. Nautiloids are the outlier of cephalopods within the octopus species. Certainly the vampire squid sits there. Now we're talking about ways that octopus are able to defend themselves, traditional octopus. Again, if you've seen the video after it's linked to you, you've seen the vampire squid. Not a lot of natural predators. Again, I'm going to bring up the anglerfish again because I think that's an analogy everyone's familiar with. When you think about that glowing light, animals like vampire squid aren't going to be attracted to that. They don't need to consume food down there. They're not out there hunting. They're drifting along. And again, vampire squid from hell does have the symbolism name-wise of a vicious predator that lives down there, but they can basically float. They're living filters that are helping our oceans by picking off detritus and doing their thing. When we get more towards a sunlight zone, again, where light is going to be represented, we get to the ability to color change for defense mechanisms. I hope you guys know the term cryptic or aposematic coloration. We're going to talk about those before we get into autotomizing limbs. But cryptic or aposematic, anybody know those definitions and why they are relevant, why they are both relevant to this animal?
So cryptic coloration means basically camouflaging into your environment by means of changing your color. And then aposematic coloration is more of a defensive coloration, where and you see it in a lot of other animals all over the world, but just basically where your color, or well, specifically these color changing creatures, you use coloration as a defense, saying, hey, don't come near me, I'm dangerous, or I'm a threat, etc. But yeah, both are super important to the blue-ringed octopus because they use both simultaneously within the video. They I'm sorry, did you want me to talk about the video too?
Yeah, sure, go ahead.
Okay, sorry about that. So what's really cool about the blue-ringed octopus is that as they're moving through their environment, they're able to utilize both. I kind of think of it as playing a first-person shooter game. Sorry for the reference. I don't know if I'm allowed to use that reference. Where you're going from one barrier to another, and you don't really know where the threat is going to be, even though you have kind of a little map telling you. And so while they're going from one barrier to another, or one place where they can camouflage to another, they're expanding and contracting their chromatophores that are lined with blue rings from their photophores, and they're utilizing that light to enhance the pigmentation and to dull it back down when they're trying to become more cryptic. So aposematic, I'm a threat, bright blue rings, for example, and then cryptic, I'm in a safe place, I can hide here. Or maybe there's food and I don't want them to see my blue.
Yeah, perfect. And excellent explanation of how easy the transition is. And we just don't see that anywhere else. We don't see the color change occur like we do here. A lot of people, I think, when we talk color change, go to the idea of a chameleon. Not even close. You'll find that out in herps. Chameleons are not cryptically colored. That's their default color. To go from cryptic across the body to epistematic, it's a feat of astronomical proportions. Most of the time, your animals out there that can perform both have usually a coloration on the underside of the body, maybe the inside of the mouth. After this, Google a sarcastic fringe head. You'll see what I'm talking about, where you have an animal that looks very basic but has to perform a behavior like opening the mouth very wide. and becomes very elaborately colored to say, hey, I tried to blend in, you were messing with me, here we go, let me show you this, see what you think of that. These animals are able to literally, like, at the blink of an eye, switch through these amazing color changes. And it's all thanks to, again, that huge complex system of chromatophores, iridophores, photophores, you name it. They've got all these tools at their disposal. As if that wasn't enough, let's add on autotomizing limbs. This is going to be important to remember, especially in a little bit here. Autotomizing, it means the self-severance of, able to remove a limb. And these limbs aren't just going to be food for fodder. They're going to be alive, moving, very, not vocal, but very, I'll just say vocal. Vocal in the evidence that they either want to be consumed or distract the potential predator. They're not just, hey, here's like, it's like throwing a steak to an attacking dog and hoping they eat it. This is a completely different ball game. Autotomizing the limbs, and then on top of that, we need the ink sac. It's a conjunction of the ink gland, the sac itself, and the sac duct. And excuse me. Again, as we move further down the water column, then we sprinkle in beautiful glittering lights as well, like a firework going off. As far as the natural energy it takes to make up this ink gland, we do have some unique things here. You've got to figure if you're pigmenting your ink cells, that's deriving your body of nutrients. It's energetically inefficient to make the ink darker than it has to be, if that makes sense. And so we see a weird phenom occur. The closer we get to the surface, I'm talking barrier reef, like you can see it, then it makes sense to have black ink. The second we start stepping down as far as light levels go, it gets much more to a red hue. There's no sense in concentrating extra energy and nutrient devotion towards making the melanin in your ink black when a simple red will suffice. Hopefully that makes sense. We don't need as much melanin as we go down in the water column because the light is different. No sense in devoting extra nutrients to pure black ink when you're in half black environment. It's devoid of sunlight. So we don't need all that extra energy devotion. Again, as we talked about earlier with the funnel itself, you can literally direct exactly where you want that ink to go and then go back to sucking in water and moving away. So at this point, if you don't think that these animals have everything going on, I don't know what to tell you. We heard about the ways that they're able to change color back and forth, blend it in, or if you do mess with them, here's a cool color change to distract you. Then I can throw my arm at you to distract you. And if that doesn't work, I'll just shoot a cloud of ink at you and then swim away. The ink for a long time was thought that it just blurred the vision. It does not. It is an extreme eye irritant. Animals don't swim out of the ink cloud to get away from it, like to still find their prey. They swim to just survive. They lose their sense of smell more often than not. Their eyes are extremely inflamed and irritated. If an animal were, like, this would never happen, but let's say we're in a closed system aquaria and you put in a 100-gallon tank a small shark and an octopus, and the ink were to stay in there, it would most definitely paralyze / incapacitate entirely the animal. It's that much of an irritant. It's like an allergic reaction. So it's that effective. These animals have a lot going on for them. This is definitely not test worthy, but I think it's really cool. So of course I'll talk about it. And if you've ever had the old Crayola box when you were a kid, you probably saw the cool little label on there that said sepia. That actually comes from a cuttlefish species. Just a cool tidbit to add. But again, if you picture, maybe you have them in front of you now, maybe that's how you're taking your notes, and I approve, especially for this lecture. But if you're using your sepia right now from the Crayola box, you'll notice it's more of a red than it is a brown, and that's truly from a cuttlefish species. So that should tell you how quickly the coloration changes of that ink pattern. Let's get back to something we alluded to earlier. Reproduction for these animals kills them. After they reproduce, males have a very short time to live. Females die after the eggs hatch. She basically Charlotte's webs it, if that makes sense. It gives it her all and then dies afterwards. We're going to find out, however, that's not the case. Let's get into reproduction first and, of course, see some gratuitous cute baby photos. There you go. I'll let you marvel at the adorable babies. Everybody loves baby pictures. Interesting here on the top picture, how the quote unquote, we're not going to call them a nest because it's really just a rocky outcropping. They don't build much of it, but we'll call it a nest for the sake of lecture today. It's interesting. You can see the behavior here, how the female is arranged around her nest of eggs. You've got to figure she has her funnel and her siphon moving water in and out, and then the arms to also circulate water. Again, you got to picture eggs in this scenario. They're not hard-shelled eggs like we would see on land. They need constant water flow. They're gonna need oxygen. And being almost trapped, if you will, inside of a cave, the mother does need to stay there to basically aerate her young, her offspring. She needs to, if this is a, hang on, let me switch colors here. If this is a cave that looks like this, so a closed off cave system, there's not a ton of water that flows through here until mom sits here, siphon, in current, X current, pumping water through, flapping her arms about. That's how we get this nice water flow to make sure the eggs are aerated. And so she becomes the vital part of their survival, doing the influx of aerated water, outgoing of waste products, again, which the eggs can do because they don't have an externalized shell, and really becomes the not only guardian, but life force for these eggs. When the eggs do actually hatch, the female is basically going to use the same method to shunt the eggs out of that den or burrow, nest, whatever we want to call it. Eggs are hatched, they look super adorable, and then mom immediately says, see ya. There are a few species that will, I won't call it parenting, but basically not shove them out of the nest early. But that's about all you get as far as parental investment here. That's kind of the extent. The larvae, once they do enter the water column, are going to basically float along in a platonic cloud and feed on very small copepods, things like that. Again, I know I've said this, but I love the term plankton because it's so vague. Plankton's just a bunch of microscopic stuff. And you got to figure while this microscopic stuff is up there, they're doing things. They're all trying to grow and survive. And when you have again, adorable, scientifically proven, an adorable small octopus in that platonic cloud, they're going to be able to do a lot more. They're going to eat larval crabs, larval sea stars, larval lobsters, like whatever's been released, fertilized gametes, heck, unfertilized gametes, single cell, multi-cell organisms that have started to divide and hopefully conquer until this baby octopus came along. We've got this huge, ever drifting cloud of larval organisms that is constantly moving through the ocean. And then we've got blue whales swimming through that and consuming a bunch of them. But this is what makes up our oceanic ecosystem. Ah, there it is. Arguably nature's greatest animal. So let's get on to the intelligence of these animals. There's, when I say debated, we all agree that octopus are intelligent. The idea of training isn't new, but it's relatively new to the zoological environment. And now we're starting to discover, wow, like we done messed up. These animals are smart. Their ability to possess short-term, long-term problem-solving skills, play behavior, and distinguish puzzles is astounding. And again, we didn't recognize and don't knock former zookeepers, we just didn't know. Enrichment and, again, the training and behavior part is new-ish to zookeeping, and certainly newish to invertebrates. When enrichment did come about, it was, of course, for animals like elephants and things like that. And I'm not knocking enrichment. Please don't take it that way. We definitely need that. That's, I think, the biggest part of zookeeping is enrichment and training and all that. It's not a minor. It's not something I want to pass by easily. But it took a bit for us to be like, hey, we threw an elephant ball in. Let's try something with the cephalopod. And then, holy cow, our lids were blown off. Their ability to figure things out is just astounding. With the cute baby photos we saw earlier, those have to come about somehow. And when octopus couple loves each other for a long time, actually it's like about a minute because they're solitary their whole lives. But when they do get intimate, we have a very special tool utilized. It is called the hectocotylus. And it's, again, if you get into the husbandry aspect, hands down your easiest way to recognize whether you have a male or female octopus. The hectocotylus is a single arm on the octopus that has, instead of the typical suckers we see, has specialized fingers on it. This is going to be used to produce spermatophores and move them down. So if you haven't heard the term spermatophore before, here in the mantle cavity, the gonads are going to produce a sperm packet, and that's literally what it is. It's a enclosed. It's like if you've ever gotten like slippers or shoes, like that weird salt packet that's in there. It's quite literally like that. It's a tiny packet containing all your reproductive gametes, and it's going to move its way down the hectocotylus. The male is going to have to insert this directly into the female's mantle cavity. We don't have broadcast spawners here. We're not going to release sperm and egg and hope they meet. We've got a special delivery of spermatophore directly into the female's mantle cavity so that reproduction is guaranteed to occur. Let me go back to, sorry, I clicked trying to get the drawings off. This is where autotomizing limbs come in, the self-severance of a limb. In multiple octopus species, that idea of being solitary your whole life really holds true here. Male octopus, more often than not, will use that hectocotylus to walk the sperm packet down to basically the distal end of the arm. So you've produced a sperm packet, you use these specialized fingers to walk it down here, and then you will rip off your arm and throw it at the female, and she can insert it into her cavity herself. That's how isolated these animals like to be. So for breeding, you literally rip off your own arm, throw it at the female, and say, don't call me. It's a very strange breeding style, but very effective for these solitary animals. As far as how that affects parenting, clearly there's not a bond here. They're not learning from mom or dad. They're not gaining any knowledge based off hunting patterns and behaviors. And that makes everything they do instinct-based, which really highlights what these animals are able to do. The instinct-based learning is incredible for a myriad of reasons. It does, of course, to us mean that you can have a baby that's never learned how to escape from a cage and does. The way we house these animals has to be like bulletproof. Nothing can go overlooked, and that makes it really tough, especially when it comes to filtration and the fact that you need a lid to get in there and feed. The enclosures we keep these animals in have to be really well thought out. They've been observed climbing onto boats, climbing into various aquaria. And again, another great example of this is right here with octopus species that were trying to figure out what went on. And another great example that's happened in aquaria. Could somebody describe what happened in this video?
So in the video, there's an aquarium that had, I for some reason thought that they were going to cohabitate some GPO and some spiny dogfish sharks. And originally, the idea was, we're going to feed the sharks. They're not going to need to eat anything. If for some reason they were to try to, mess with the octopus, it'll be okay because the octopus is just going to camouflage. Maybe they'll, run and hide. It'll be fine. And all of a sudden, the sharks kept turning up dead, and they couldn't figure out what was going on. And what they ended up recording and figuring out that the octopus had decided to basically hang out and kind of hunt the shark, waits for the shark to come up above and grabs him and flips him over. And in flipping the shark over, kind of hits this tonic immobility. The shark is going to go into almost like a trance like state. And basically, the octopus doesn't really need to defend himself because the shark is in this trance, can't really do anything. And the octopus is just killing these sharks over and over. And the aquarium really didn't think that through, or just kind of thought like, oh, the octopus will defend itself, no big deal, but never thought like, oh, it's just going to flip these sharks over and just kill them.
Perfect explanation. I'm so glad, too, that tonic immobility was brought up. Because here's what's interesting, kids. Yes, it was, again, don't blame former zookeepers. We just didn't know octopus were so smart. The idea that, and again, they didn't know why these sharks were being winding up dead. There weren't like a ton of sucker marks or beak imprints or chunks taken out of them. This, I hope, highlights, however, what makes these animals so intelligent. Using tonic immobility to put I don't think I could phrase it any better, a shark into a trance-like, almost, if they're in it long enough, coma, basically, in order to incapacitate them. You never learn that anywhere. You're only 3 years old. It'd be like your kid, like, doing college algebra at 3. Like, where did you learn this, young friend? These animals are so intelligent. The fact that they're able to learn, hey, instead of, I don't have to fight the shark, I have to flip it upside down and then hold it there. That's end game. That ability to solve these problems, and yes, unfortunately, a lot of the times the problems are how to incapacitate preys or threats very quickly, really resonates with how incredibly intelligent these animals really, really are. Now to the moment you've all been waiting for. Oops. The short lifespans. I have my own conspiracy theories. I'll talk about them, but I do truly think that it's no coincidence, the fact that these animals live such short lifespans. To have a GPO that, again, as was said earlier, literally detect a threat in their tank and then wait till the night janitor moves along, goes out, and instead of attacking a shark, just says, hey, I'm going to flip you upside down for a bit, bud. There goes my threat. Now I'm set. Taking out dogfish, shark left and right. This ability to learn and grow and compound, and again, like all the lessons your parents taught you growing up, like every mistake you can learn from, these animals do that times a million. Their ability to grasp what's in front of them and solve kind of life's puzzles is astounding. It's literally only limited by their lifespan. With that lifespan, we have a crazy phenom that occurs, and it's an genetically programmed death. If we remove parts of the endocrine system, this, maybe it's a phenomenon, maybe it's like the saddest thing to happen to nature ever. But even if the endocrine system is removed, some stuff still happens. Anybody know what those are?
So when the endocrine system is removed, it's only been shown to prolong I can't remember if this specific example was like a breeding or a non-breeding individual, but anyways, it only prolongs their lives for just a few months.
Yeah, so even if the endocrine system is removed, um, the processes that occur here are just insane. Uh, all right, where do I begin? The idea that we've studied these animals long enough, had them in captivity, know they're learning. Imagine your favorite animal that you've been training, working with for so long, and then something comes up. Uh, there's so much that happens here. The processes that occur aren't always medical or veterinary intervention. They're going to have adaptations that change. Um, yeah, based on behavior more than anything. Does anybody want to highlight that?
Yeah. So, sorry. So talking back on the endocrine secretions, um, when an octopus matures, their like, as any other organism, they're run by their body's hormones, chemicals, etc. Basically just neurological instructions. And once they mature, their decision their motive is to breed, reproduce, pass on their genetics. So once they've done that, they go into this state of, it's almost like how hormones change in humans after having children, or big life events. Their endocrine systems stop producing as much of what kind of motivates them. And I apologize for not having the correct terminology or labels, but yeah, so the endocrine secretions become kind of more minimal in substance, and it almost creates a type of uh I can compare it to, I'm sorry, suicide slash dementia. And I apologize that that sounds kind of crass, but I mean, these organisms, they literally just even with keeper intervention, they will ultimately just die. So um with the suicide aspect, the males, after breeding, and um this is really insane to me, because I always tell literally everyone that if octopus didn't do this, they would probably be like, up there or even above us. Like, humans would have to answer to them, because their ability to learn, like, is just so intense, and they're so intelligent and can fit through small holes. But anyways, so once a male has bred, they will, typically both male and female stop eating. Again, with keeper intervention, you can try to force feed them, tube feed them, whatever. uh, Ultimately they'll stop digesting, and with not digesting, they're not able to function anymore, and your whole system shuts down, and you just eventually stop running and being, uh, hence death. So but with males, they'll actually display, like, make a spectacle of themselves, rather than using their aposematic coloration for warning, and their cryptid coloration for camouflage. They'll go out and start just flashing colors, become less fearful of predators, and literally begging to just, eat me, take me, because if they're not shutting down fast enough, or if their system's not doing what it's genetically programmed to do in nature, then that's when they start. Again, they just start swimming out in the open, flashing their colors with, I mean, light provided. Um, bioluminescence, whatever the tools that they have, and they're just ultimately programmed to just die after reproduction. Females will lay their eggs, and then they basically sit there for until their eggs hatch, and then once their eggs hatch, it's okay to let go for them. It's really sad, but it's really actually interesting, and also terrifying to think what would happen if they didn't do this.
I am just so happy that my weird conspiracy theories have resonated now through the halls of eternity. Um, I really do. I'm sorry, but I think it's true. Uh, these animals have a genetically programmed death. For a lot of mammals, for example, genetically programmed death means usually dentition problems, problems with the teeth either getting decayed, falling out, rotting, and then the animal can't eat. It's nature telling you to die. Elephants, for example, aren't really known to get cancer or any other illness that necessarily incapacitates them to the point of death. Their teeth fall out. And is nature saying, hey, you had a good run, like time to go? Here we have nature saying, whoa, this is too good of a run, time to go real early. As was mentioned earlier, and it's spot on, the idea of all the interventions we've tried to pull. Again, I know it was said, I'm just going to reiterate it. The idea that, again, think of Charlotte's Web. The female octopus is staying, tending to her nest, not eating. She's going to starve to death. We intervene surgically and genetically, remove part of the endocrine system that programs this death. The female still doesn't eat. Okay, what's our next step? Force feed. Okay, now all of a sudden the food passes through the body and no nutrients have been absorbed. Like, there's no explanation for it. And 100% everyone, this is a complete conspiracy theory. It is not scientifically proven, but I kind of think it might be true. If you were to allow these animals to live for, say, human lifespans, they would definitely be our overlords. Like, picture your 3-year-old being able to solve complex puzzles and camouflage itself. Like, that's horrifying to think about. It'd be like you're learning, like, I'm not joking, intelligence-wise, it's like learning your alphabet on day 2 of life. The neurological processes that are occurring are second to none. Everything that was said earlier was just like spot on. And again, I want to reiterate, complete conspiracy theory that they would run everything, but I believe it. They are. Nature has I'm sorry, but nature's been doing it for a long time. They have a pretty good checks and balances system. If nature were to allow an octopus, like let's take a GPO, we're allowing an octopus to live for 20 years, much less 60 years or 80 years. Like, the fact that they keep learning and compounding on the data, they're living supercomputers with what they're able to do. And that's just speaking about what you guys know as intelligence. The reason I say that is because literally only 1/3 of that brain that makes them so amazing is located where we think the brain is. 2/3 of the other neurons are located in the arms themselves. They're distributed out through each arm. This serves, again, I told you earlier to remember the term autotomy, the self-severance of a limb. Now we're moving on to autonomy. Make sure come test time we understand the difference between the two. So in autotomy, self-severance, autonomy is individual function. With autonomy, I'm trying to think of a nice way to say this without blowing my mind with how cool these animals are. It means that the arms don't, I don't want to say that they have their own brains, but they really do. This animal is extremely complex with how it's laid out. The one weird thing here, not that everything's been weird so far, which was what makes it amazing, is the brain being only 1/3 the size of the other quote unquote brain, which is again a cluster of ganglia inside of each arm running through the whole thing. The 1/3 brain compared to the 2/3 brain can only issue commands to the arms. This rises in some strange learning behaviors. One of which being the fact that in order to issue this command, the brain has to say, do this. And then in order to make sure the arm's doing it correctly, it has to visually observe it. There comes another strange quirk with this learning pathway that these animals are taking. This is kind of like the new math curriculum in schools. Like, It's a new way of learning, but in this case, it actually works. Does anybody know what else would come with this idea of the neurological pathways that are running?
So the way it works is like each individual arm is kind of capable of learning its own thing. And I always kind of think of it as like, and this might be weird, so stay with me, but like when you're learning to drive and you're really focused and you have to like watch everything you're doing, and really the only feedback you know is like you're watching and you're paying attention to what's going on. But like once you get comfortable and you're good at it, you know, all of a sudden you get in your car and you're driving to work, and then you find yourself at work and you've just kind of accepted this muscle memory of what's going on. I always think of it like that, where they're watching their individual arm telling it, okay, we're going to open this jar. So they watch, they figure out how to open the jar. Now they're good at it. That arm is now the jar opening arm. That muscle memory is good. The octopus gets a jar and it's like, okay, you know, arm 3 over here, where the arm opening or the jar opening arm, do your thing, because it's now just accepted that that's the muscle memory for that specific arm, and it can now go through each kind of appendage that it has and work out individual muscle memories, and they're all going to learn independently because there is no like feedback going to the brain. It's just what they're choosing to do with it.
Oh my gosh. Never in this class ever apologize for weird analogies. That's what we do here. Analogies are my jam, and the weirder the better. And it really, I think the best term there was muscle memory. And now instead of muscle memory, we're getting muscle memory plus learning. So let's take what we just heard from what we previously heard about octopus overlords, and now the fact that their arms are learning on their own. That's all very true. We've got it, and I'm not going to say the most intelligent animal on Earth, obviously, but the learning curve is hands down the most intelligent animal on Earth. From point A to point B, and then the unfortunate, or maybe fortunate for us, I guess, unfortunate death of these animals, the learning curve is out of control. That's just talking about the brain. Now, from what we just heard, now we're talking about the arms learning individually. The jar opening is a great example. An animal that's going to be able to open a jar, and then that arm individually is going to learn to be better at opening jars. That's astounding. The only, and this is a very small asterisk, the only odd thing here is, again, that neurological pathway back. We've got all these great things sending out. Brain says, hey, there's a jar, I want to open it. Arm does it, brain says, good job. I have to observe that visually to make sure the arm did the job. And then I say, good job, arm. Arm comes back and says, I got this from now on. The brain is never going to have to tell that arm how to do that from here on out. The first time it has to be visually confirmed, brain says, good job, sends it to the arm. Arm's going to learn from there. Again, the only detraction here for points, like this is like a perfect dive and you're giving them a 9.5, is that there's no neurological pathway back. This is why everything has to be observed and confirmed the first time. From then, arms can take it. The pathway back, however, that part's a little weird. This is the equivalent of, and please don't do this at home, this is not a recommendation to see if you're an octopus, one of our octopus overlords. The only analogy, though, is the idea that you would have to put your hand on a stove burner and then pick it up and look at it to see if it was burned. You're not going to see that pathway and the feedback from the arm itself. You have to visually observe it because there's not the connectivity back to the brain. Again, it's a minor sly on these animals for what they're able to do, but it is an interesting phenom that occurs. And it's more than likely because 2/3 of the brain are in the arm. The brain doesn't get that feedback. So now we've got, again, not the most intelligent animal on Earth, but timeline-wise, the most intelligent animal able to solve complex puzzles at a very young age with no parental representation. And I think that should be brought up too. The fact that the parents aren't involved. A lot of your, let's say our intelligent animals, let's take apes, and elephants, for example, what's one common theme that runs amongst them? Parental investment. You don't see elephants being just and if you've ever seen an elephant birth, it's not pretty. But you don't see an elephant just dropped basically on the ground, and mom walks away and says good luck. Uh, Males and females both stay with the herd until males become sexually mature, then are driven away, and then we've got this huge, amazing matriarch of mother, aunt, grandmother elephants that lead the way and teach their offspring. Apes the same way. You don't see a lowland gorilla give birth and then throw their baby in a hammock and say good luck. Uh, they're constantly hanging on one another, seeing each other forage, learning what foods to eat. There's so much parental investment. And finally, we'll get to humans. If any of you out there have children, and this is not an advisement to have children, my two are thankfully being quiet upstairs right now. Think about your child if you never spoke to them and just left them in their crib, and then you walk in there 3 days later and they're starting to do calculus. Not going to happen. The learning curve here is astounding with what these animals are able to do. Again, the only weird part here is that, let's put it this way, we'll use kids. Again, you walk in on day 3, your kid's doing calculus, but then your kid can look at you while his hand just does calculus because that arm is learning on its own. The second you realize 2 + 2 is 4, and I know it's not calculus, but it's as far as I know in math. I'm a zookeeper, not a mathematician. He's doing 2 + 2 + 4 and saying, Mom, this is calculus, while he's looking at you in the face. That arm's going to keep doing more complicated processes, like maybe 3 + 3, which is as high as I know. But just think about that. You've got an animal now that's intelligent, learning all the time, and then on top of that, each arm is creating new neurological memories. And again, back to the driving analogy, new muscle memories and getting better at them, not just learning it, perfecting it. From taking down a crab, I've learned this time that I got pinched. I should rip off those claws first. And each arm is getting better as it goes, along with the brain. It's an astounding compilation of knowledge. I'm going to try to go through the rest quick. I realize I've been yelling about octopus for a long time now. I love them. They're amazing. Hopefully you do too. Back to all these color changes. Now we have a compounding animal here with the mimic octopus. The mimic octopus is extremely unique. Not only is it able to do all the things that octopus are able to do with the color change, now we have a behavioral change in order to mimic other sea creatures. They're about 2 feet long, but their behavior pattern is extremely unique. Again, what is... All right, a couple interesting things here. First off, the species isn't discovered until almost the year 2000, which is crazy because mimic octopus have been around forever. They've been photographed and documented. Scientists actually went back and found a bunch of photos where people thought they identified, say like the picture up at the top right, a sea snake, when really it was a mimic octopus. It's so good at behaving like other animals that we didn't even recognize it as a different animal. I guess it's the biggest compliment you could pay to them. They're very good at what they do. There'll be a video link in your notes that shows the varying behaviors they exhibit. What is interesting, and again, I'm sorry to stop here. I said I was going to go fast. Turns out I'm not. What's interesting about these animals is the fact that, again, with no parental investment, so it's not like they learn from mom and dad how to look like a brittle star or a giant crab. They didn't learn that. They, through their lives, pick up on what seems to be a dominant species or predator in their environment, something threatening, and then learn to imitate that. The most interesting part about this is, and this is not a regular occurrence, but it has been observed in captivity with the very few facilities that keep mimic octopus. Again, I hope this puts into perspective how smart these animals are. Coming from an environment where there's no natural predators, let's take all the variables out of the equation here. Let's say you bred a mimic octopus in captivity, took the offspring the second it was pushed out of the nest, if you will, put it in an individual tank, it's going to learn from things around it. It's not going to mimic a sea, let's say a sea snake, because there's not threatening sea snakes around it. Hopefully you're running a good aquarium. There's not a big threat to it. It's been observed with captive mimic octopus that they pick up on things that are relevant to them in that environment. The notable experience I'd like to talk about is when the animal was fed. It was usually, of course, most of them are nocturnal. When the animal was fed, it was always at night at this particular, it wasn't an aquarium, it was for science studies, but the keeper and the janitors would walk by, and the thing that the animal started associating with food was the movement of keys. And every time anyone would come by the aquarium, exhibit, excuse me, anytime someone would come by the exhibit, the animal would shape itself to mimic keys jangling. That's what it thought was the dominant thing. That's what provided food, took away food. And just that intelligence in that very, very short lifespan. Mimic octopus do not live long. The ability to pick up on the fact that, hey, keys are the dominant species here, should really highlight how intelligent these animals are. All right, now I'll try to go faster, but it probably won't happen. A couple pictures of mimic octopus highlighted here. They're able to do amazing things, especially once they burrow. They're notorious for digging small burrows, putting essentially the mantle, or what we would consider the head, down. So they do appear like just a cluster of rays and arms and other scary aspects to them. Enough about octopus. Let's get on to our notable mentions here with the squids. And again, ton to learn about squids. We don't have enough time this semester. I could spend clearly more than a day talking about cephs in general. And they're just they're a cliff note, an excerpt on mollusk in general. But we do have to give credit where credit's due, and that is to the giant squid. It is, without a doubt, and hopefully I'm wrong, I do want to be wrong on this, the largest invertebrate on Earth. I hope one day we discover things even bigger. Like I said earlier, their sizes are grossly exaggerated. Again, when they wash up on shore, it's 100-foot long, tackling submarines and 20,000 leagues under the sea. But that shouldn't diminish how big they actually are. They're huge. A lot of that, of course, is built up off the tentacles themselves as far as length. But colossal squids versus giant squids definitely take the reins as far as mantle size. The environment they live in obviously is at a detriment for us to study them. A lot of what we know about them comes from these lovely animals, the sperm whale. Sperm whales, as you can see on the bottom right here, their faces, and trust me, if you guys all vote for it after this, I would love it. I always think that we should do a cetacea class where we talk about whales. I could spend hours on hours, not that you'd want to hear it, but I would do it, on what's called the spermaceti organ. It's what allows sperm whales to dive so deep and then come back with no sensible detriment to the animal. Again, you're talking about a mammal here that has to come up and breathe air, but able to dive incredibly deep, apparently tackle and giant squid are one of their primary food sources. That's really where we gain a lot of our knowledge about giant squid, unfortunately. So very cool animals. Do we know everything about them? Not even close. We know a lot of what we find in stomach contents of sperm whales. We know about that there's some intense battles, but yeah, not a ton known. Here's our first image of a, for now, live giant squid. In 2006, that's the let me go back. This squid initially gets off the hook. In 2006, we finally capture in Japan the first quote unquote living giant squid, and the animal died shortly thereafter. If you have an animal like this, why in the world would it die after a capture, aside from being like dragged on the hook? Well, we do that with fish all the time, right? We hook them, you can take the hook out, throw them back in the water. what, aside from the capture itself, would cause an animal like this to perish?
These animals are structured for their environment, and giant squids are typically found in the deep sea. Um, and so when this one was hooked up, or anyone was hooked up, uh, it's almost like why we need a spacesuit in space. So you're pulling this animal out of its highly pressurized environment, and it's beginning to expand, which means that they're actually bigger than what they would be in their deep sea ecosystems, um, as you're bringing it up, and ultimately it dies because just the pressure is too, it's not enough for what they're structured for, and their organs and everything inside it expands basically. But I love the analogy with, like I said, with needing a spacesuit, because in space, if we went into outer space, or even certain levels, like, uh, towards the atmosphere, we would start to like implode because the pressure is so great. We would die, and there's no way, everything would collapse within itself, and we wouldn't be able to breathe or anything, think, whatever. Everything would just like snap into like one. It's just really crazy to think about. So it's actually the opposite for these guys. Any deep sea creature that is pulled up has died, if not by the hook, on the way up. It's died because, again, it's like the space analogy, but opposite, where they're just expanding and they can't function anymore.
I love the analogies we're getting here today. Muscle memory, driving to work blind, pretty much. Please don't do that. But the feeling of getting to learn to drive, and now we're getting another analogy of reverse outer space, and honestly, that's as relatable as we can make it. Um, I don't think there's a better analogy for it as far as stuff that we get and we can comprehend. Uh, this isn't anywhere close to like the bends, which if you've been a diver or taken scuba classes, you know all the dangers of that. Uh, it's not even close. It's not a comparison there. With the bends, you're like, and I'm sorry, I'm not up on my stats. I think humans can get the bends from like 60 to 70 feet down. It might be more, it might be less. Don't at me. Um, but we very quickly suffer extremely negative effects from that rise up to the surface versus the pressure we were feeling. Um, and surely you've seen some movie where people are deep sea underwater and have to slowly make their way up. Now imagine not only the injury from the hooking itself, but now imagine you're being just thrusted and yanked up from the bottom. And again, I think reverse outer space is the best way to say it. There's also one other factor that figures in here. A giant bodied organism like this is almost living in outer space. Um, you're able to allow water to control the huge gelatinous matrix of your body. Literally being brought up to the surface is you're going to be crushed under your own weight. It's why, like, let's say you go underwater, or you're like, let's say you're in the shallow end of the pool even, and you can lift somebody that's just as heavy as you up quite a bit. It's because water is controlling the rest. Um, like you have this colossal body, and all of a sudden this huge biomass is being dragged onto a boat. You're literally crushed under your own weight. Aside from the reverse outer space analogy, aside from the hooking, the huge drag up, now your body can't sustain that enormous weight sitting on the edge of a boat. A lot of complications occur, obviously, but I don't want to encourage it, but good to see a giant squid. A couple of quick claim to fame before we get done today. With the giant and colossal squids, largest eyes in the animal world. Again, everybody, when I say stuff like this, there's a reason. There's a reason it's highlighted. Why would these animals have the biggest eyes, the largest eyes in the animal world? You're living in a completely light devoid environment. The second you see a sparkle, let's go get it. It makes sense to make do with the low light environment to start developing larger eyes throughout evolution. That way, the slightest little tick, the smallest little glitter sparkle that happens, I'm on it. Let's go. It's again, we don't know enough about these animals for me to say this confidently, but probably why they have grown to such immense size. Being able to detect all these, again, we'll use the anglerfish. Oh, cool, there's a little light sparkling. I'll go eat that instead of it being a prey lure. Now it just tells me where the food is. So again, I can't say that confidently science-wise, but they're making do in a world devoid of light. Here we have the difference here, in case any of you were shouting at your computer screens earlier. Difference between colossal and giant squid. Giant squid is significantly larger as far as length. The mantle cavity of the colossal squid, it's beefy. Colossal squids are significantly shorter in length, but much heavier, if you will, in girth and biomass. Again, one of the things you see here, let me get these red arrows off the screen. One of the things you'll see is tentacles. This is what gives them their length here as well. Interesting with the colossal squid, they're going to lack the hectocotylus. They have a method of reproduction that it's what we think happens, but we won't even mention it as far as test questions here because it's not confirmed. Colossal squid, though, just bigger mantle. Giant squid are just crazy, crazy long. Again, like you're talking 60 feet from the tip of the fin to the end of that tentacle. But again, 8 arms. And that concludes the amazingness that is the cephalopod group. I hope you guys enjoyed it and have a new perspective on again, these primitive, simplistic animals and what they're really capable of.